After going through this module, you shall be able to know the difference between human and non-human bones, different distinguishing parameters between humans and other mammals. Additionally, you will be knowing how non-human animal bones are commonly confused with human bones. Distinguishing between human and non-human bone is a task that should only be undertaken by a forensic anthropologist or individuals experienced in osteology. The purpose of studying this has twofold importance. First, it is included to demonstrate that non-human bones can easily be mistaken for human bones and to reinforce why an experienced osteologist should be making the final determination. And second, this module is also included for forensic anthropology students to introduce them to different examples of non-human bones that can be easily confused with human bones. There are several questions that the experienced forensic anthropologist or forensic archaeologist should be able to answer in the field. These include determining whether the suspected material is actually bone, and if it is bone, then is it human or non-human. For example, rocks will appear in shapes that can mimic human bones. One rule of thumb is that rocks are usually heavier than bones. The irregular bones that could be mistaken for rocks such as patella, carpals, tarsals tend to have a higher concentration of trabecular or cancellous bone, making them significantly lighter than rocks. An experienced forensic anthropologist will have no problem in distinguishing non-osseous material from bone. More commonly, the forensic anthropologist will be asked to determine whether the skeletal material is human or non-human. In many cases, being able to differentiate human from funnel bones enables the investigator to eliminate what may have been at first considered to a forensic case. And William Bass, who is a noted forensic anthropologist, has stated that as many as 25 to 30% of all the cases submitted to forensic anthropologists for identification are non-human in nature, and many of those end up being butchered domestic animals. Thus, basic knowledge of the human skeleton and non-human skeleton can help to save time when determining the forensic significance of whole or fragmentary skeletal remains. Of course, as a rule of thumb, all bones recovered from a scene should be examined by a forensic anthropologist, since such an expert will have the expertise to determine whether the bones are humans or non-human. If you do not have a forensic anthropologist on the scene or readily available, one suggestion is to take digital photographs and email them to a forensic anthropologist. Many forensic anthropologists have reviewed photos via email to make the determination of human versus non-human bones. This can serve as a tremendous amount of time and resources. Next, you will be knowing the cremation or thermal damage to skeletal remains. Cremation is basically a process that uses intense heat to rapidly reduce a body to ashes and small bone fragments. Bodies are typically burned prior to burial or within the grave and may be scarred or partially, incompletely or completely burned depending on the intensity of fire to which the body was exposed. The remains may be mixed in with other elements of the burial, left as a surface deposit or held in some type of container. In a forensic investigation, forensic anthropologists examine cremated remains originating as a result of criminal activity and also as a result of normal cultural practices when a loved one dies, in the case where remains are cremated as a result of the perpetrator deliberately trying to dispose of the remains. The remains will most likely range from charred to incompletely burnt, as it takes extremely high temperature to destroy bones. Almost impossible to completely destroy all evidence of a body through burning. Because of this fact, cremation that result from crematorium are normally reduced to very small particles depending on the type of processor used to reduce the remains. Now, I will be describing how to distinguish humans from other mammals. Large mammal bones such as bear, deer, dogs and pigs are most often confused with adult human bones, and small animals may be confused with the juvenile or fetal bones. In particular, when bones are highly weathered, eroded and fragmented, identification may only be possible when conducted by a trained osteologist. From an anatomical perspective, humans and other non-human mammals can be very similar in their skeletal components, and because humans are mammals, they possess many of the same skeletal characteristics. For example, the number and types of bone are similar among mammals. In addition, key features such as bone structure are similar, so that the most mammals have long bone shaft that are composed of thick cortical bone and long bone ends that are composed of trabecular bone. In fact, if just a small bone fragment is recovered without any morphological indicators, there is the only way to identify whether it is human or non-human would be through histological examination or DNA analysis. The figure is showing you the comparison of different stages of growth of a human femur. Upon gross examination, there are two main characteristics of bones that can help in making the distinction between human and non-human bone easy and expeditious, that is maturity and morphology. Maturity aids in differentiating small non-human animals that even after reaching adulthood have bones that are similar in size when compared with juvenile humans. Depending on the level of development, juvenile human long bones may have unfused separate epiphyses or may not possess any at all. Conversely, those of a small adult non-human animal will display fused epiphyses. As a result, non-human bones can be easily differentiated from juvenile humans by examining the level of bone maturity. The proximal and distal ends of the juvenile long bones are roughened in appearance, whereas the epiphyse will eventually fuse to the metaphyses. In addition, other morphological indicators that can be useful when differentiating small animals from human juveniles long bones may include non-human skeletal features such as the fusion of fibula and tibia and the curvature of long bone shaft, that is diaphysis. For example, the long bone shaft of small mammals may be noticeably curved where it is straight in healthy juveniles. An example of the characteristics that distinguish juvenile human long bones from those of the small adult animals. One structure of small mammals that could possibly be confused with the human or fetal bone is the pelvis. If a small pelvis is fused into one unit, it will be a non-human pelvis, because a human juvenile pelvis of a comparable size is still in multiple pieces, and two adult pelvic bones of the human do not fuse at all unless there is a pathological condition. The image is showing you the pictorial comparison of the anterior view of a right newborn femur with those of small adult mammal such as a rabbit, an armadillo and a raccoon. The another diagram is showing you difference in the diaphyseal curvature between a human newborn femur and a raccoon. The second characteristic of bone that aids in the distinction between human and non-human mammalian bone is morphology or the shape of bone. Because humans are bipedal, they have distinct morphological features related to walking upright, which distinguish them from all quadrupeds that are adapted for four-legged locomotion. When comparing humans and other mammals, the dissimilarity in shape of long bones and other bones of the body, such as the scapula, can be very distinct and can quickly lead to a positive identification of human or non-human domain. For example, in a number of small animals including mammals, the fibula is reduced in size and it is fused to the shaft of the tibia. In humans, the fibula does not normally fuse to the tibia unless there is a pathology such as ossification of ligaments that serve to keep both bones articulated together. Furthermore, some larger mammal species, for example pig, sheep and deer, also have a curved and fused radius and ulna. Recognition of these two bones ultimately exclude humans as both the radius and ulna have straight diaphysis and remain unfused throughout the life. Mammals belonging to the order Artiodactyl can be easily distinguished from humans by the presence of metapodial. The next figure is showing a comparison between the human adult child pelvis that is shown in A and that of the adult raccoon in the B. Although roughly the same size, the human pelvis remains unfused in young children and the sacrum always remain as a separate unit. In another diagram, it is showing the comparison of mammalian scapula. In A, there is a mammalian scapula. In B, there is a mammalian scapula of sheep. In C, it is of adult human, and in D, it is of dog. Metapodials are long in certain species and therefore could be easily mistaken as long bones. Metapodials are easily recognized by a number of morphological indicators. The shaft are long, thin and straight, and they still retain a clear groove down the shaft where they have fused. The proximal articulation is flat and the distal articulation is unique with double rounded articulation. Furthermore, metacarpal and metatarsal can be distinguished from one another by cross-sectional shape of their shaft. Metatarsal are square shaped and metacarpals are D-shaped in the cross section. The figure is showing you the comparison between a human newborn tibia that is shown in A and the fused tibia and fibula of the rabbit that is shown in B. The human cranium is quite distinct from any other mammal because humans have a large rounded brain case and flat or orthognathic face in profile. Now, I will be telling you different non-human animal bones that are commonly confused with the human bones. There are several non-human animals that have bone structure or features that appear similar to human skeletal material. There are some bones, however, that are more commonly misidentified. Animal bones that might be confused include the bone of a bear paw, pig teeth, the knee of a pig and a turtle shell. Disarticulated bear paws are commonly confused with human hands. After a hunted bear has been killed, the claws are usually removed as a trophy and paws are sometimes discarded after they are skinned. There is a striking resemblance between a bear paw and a human hand after decomposition of the soft tissue has started. The bear paw can be easily identified by examining the individual bones, and it can also be easily differentiated from a human hand without cleaning the soft tissue from the bone. A radiograph can be used to differentiate the bear paw from the human hand because the morphology of human hand bone is clearly different. Pig molar teeth also show a striking resemblance to human molars because pigs are omnivorous like humans and their teeth are designed for a generalized diet. In addition, the proximal part of the pig tibia called the tibial plateau can be easily misidentified as a human tibial plateau by an inexperienced osteologist as they share a similar morphology. The tibial plateau of a butchered pig knee or an unfused proximal tibial epiphysis may be easily identified when a portion of the proximal pig fibula is also available for examination. However, if only the very top of the tibial plateau is available without the shaft of the tibia, it can be very difficult to differentiate from a human tibia. Conversely, the tibial plateau of the deer is easily differentiated from the human knee because of the strikingly different morphology. When the outer layer of the turtle shell that is made up of pieces called scutes comes off, the underlying shell may resemble the top of a human skull. This is particularly true if only part of the top of the shell is visible through the soil and has been bleached from sun exposure. Also, fragments of the turtle shell from archaeological context can be easily mistaken for fragments of human cranial bone because the flat shape and thickness is similar to human cranial vault fragments. If there are no diagnostic characteristics on the outside of the turtle shell fragment, then the cross-sectional morphology of the shell can be used to differentiate from the unique cross-section of human cranial bones, which are composed of diplo or cancellous bone. The diagram is showing you comparison between the human and non-human skull. The non-human skull is of chimpanzee, orangutan and macaque. So students, let us see what we have studied. Distinguishing between human and non-human bone is a task that should only be undertaken by a forensic anthropologist or individuals experienced in osteology. The purpose of studying this has twofold importance. First, it is induced to demonstrate that non-human bones can easily be mistaken for human bones and to reinforce why an experienced osteologist should be making the final determination. And second, this module is also included for forensic anthropology students to introduce them to different examples of non-human bones that can be easily confused with human bones. Cremation is a process that uses intense heat to rapidly reduce a body to ashes and small bone fragments. Bodies are typically burned prior to burial or within the grave and may be charred or partially, incompletely or completely burned depending on the intensity of the fire to which the body was exposed. From an anatomical perspective, humans and other non-human mammals can be very similar in their skeletal components, and because humans are mammals, they possess many of the same skeletal characteristics. Upon gross examination, there are two main characteristics of bones that can help make the distinction between human and non-human bones easy and expeditious, that is, maturity and morphology. There are several non-human animals that have bone structures or features that appear similar to human skeletal material. There are some bones, however, that are more commonly misidentified. Animal bones that might be confused include the bones of a bear paw, pig teeth, the knee of a pig and a turtle shell.